Okay, good morning. It's 10 a.m. today. Good morning, good afternoon, good evening to all of you. Join us today for our last webinar for this year before our holiday season. And thank you all for joining around the world, our subscribers from India, Spain, Africa. Thank you all for joining. My name is Dil Thavarajah. I'm a professor here at the Clemson University, oversee the Pulse Quality and Organic Nutritional Breeding Program at the Plant and Environmental Sciences. This program is sponsored by the United States Department of Agriculture, OREI, NIFA, and USAID Feed the Future Innovation Lab, South Carolina Department of Agriculture, the Good Food Institute, and Food Global. Thank you for all the funders for helping us to continue our breeding program, as well as bringing all these guest speakers to give us more knowledge on pulse breeding. First, I'd like to thank for Mr. Tristan Lawrence and Elizabeth Bean for helping us for the all year bringing these speakers and organizing the webinar series. And thank you, Tristan and Leslie, for all the help. And I would like to introduce one of the best outstanding professors and the pulse breeders that I have known for my career as a pulse breeder and the nutritional work that I have done globally with Dr. Diego Rubiales. Dr. Rubiales is a very well-known pea breeder. He specialized in legume and cereal crop breeding for genetic crop improvement, disease resistant. He's also a full professor at the Institute for Sustainable Agriculture, CSIC, in Spain. The other research interests including the development and selection methods, evaluation, collection, and intra and interspecific crosses. And also, Dr. Diego and we have started a collaborative project to develop cultivars that best suitable for high protein quality here in South Carolina. And Dr. Diego has published more than 350 research articles, and he is the senior editor for the Frontiers in Plant Breeding section. And also he has done so many international and national programs and a project about 20 Spanish projects. And he has been known for the best pea breeder in the world. Thank you, Dr. Diego, for joining with us and sharing your information to us. And everyone, welcome. And now the floor is yours. Thank you.
Thank you, Dil, for the nice presentation. Hello, everybody. Good morning for you, for those that are in the States. Good afternoon, good evening. Here is just good afternoon in Spain. It's just me. It's all I have is me, and not so important as Dil said. Just to present you where I work at the CSIC, that is a national institution in Spain, let's say, High Council for Scientific Research. It's not the University of Cordoba. So here we have a map where we have Spain. Cordoba is in the very south, just very close to the north of Africa. And I mark it in yellow the area of influence, the agroecological condition for the where we work. You can see North Europe is Europe is more green, it's more rainy area, and we are not as desert, but let's say some kind of semi-desertic area. We are typically Mediterranean weather, what means the presentation is not working. Now here, what means a very, very dry and very warm summer. So we can grow in summer when we have irrigation, but most of the country is rain-fed, so basically we focus on crops that we plant late in autumn or winter and we harvest late in spring. We are focusing, therefore, mainly on typically Mediterranean legumes like fava bean, pea, lentils, chickpeas, focusing for this period, planting even what others would call spring types. We plant in winter, profiting from the winter rain, and we harvest at the very late spring or very early in summer. For us, legumes are in Mediterranean basis, as traditionally, historically, has been very important. They have a key role in the animal feeding, different forage or feed components, but most importantly, they have been a base of our diet for millennia. So they are farmers or consumers, we are very used to grow and to eat legumes. However, in the last 50 years, with the introduction of modern agricultural techniques, legumes were left a little bit aside and cultivation decreased, as I will discuss later. But still, there is a renewed concern on the importance of legumes in the sustainable agriculture and the environment, and there is the interest to revert this trend. Just doing a little bit of history, I still mark in yellow the area of influence where we work. Agriculture started in different places, different continents and different times, and where different crops were grown in isolation, but always there was, in all the civilization, there was a legume and a cereal. In America, South America, Central America was maize and common beans. For us was wheat and fava bean or lentils and so on. But the important thing is that first, slowly, with boats, with the migrations, with colonizations, the crops move from one area to another. Today, this can happen very fast, but before it was very slow. So during several thousand years, pea, fava bean, lentil, chickpea were cultivated and evolved in the Mediterranean basin and then moved to another continent. But that is the crops and the areas that we are interested now, just very rapidly. Lentils today is more important in Canada, but this is, we are talking about 50 years. During ages, millennia has been in the Mediterranean, and the same applies for Phaseolus, that was typically Central South American crop, and with colonization move was introduced into Europe and from others. So, and here is the role of the plant breeders to adapt the crops that move to another region or that evolve, making them more profitable, better adapted to the different stresses, the environment, the soil, the climate that is also changing. And even we have a very opposite situation. We was talking about Mediterranean crops moving to other continents, Phaseolus moving from South America to other continents, and we have soybean that was typically grown in isolation in China for ages, and very rapidly, only recently became an international crop, and today is just the opposite. So it's the major legume crop cultivated, and it's mainly the States and Brazil, Argentina, and the major importer is China, where Europe is also, for us, is very important crop to import, but you see, our market is nothing compared with the Chinese market. Coming back just to Cordoba to present you where I am working. Cordoba is a small town, let's say about 300,000 inhabitants, with a small university, but very much focalized on agriculture. So the School of Agriculture of Animal Sciences are the dominating ones. And we have, as I said, we don't belong to the university. We have our institute and the regional agricultural institutes that are sharing facilities, and we are sometimes working in interaction. Cordoba was very important during the Roman times. We still have a Roman bridge still in operation. Till some years ago, there were even cars were passing. Now it's only pedestrians. It's a major touristic event. And Cordoba was even more important later on. Let's say this was about the Romans were 2,000 years ago, but about 1,000 years ago, in the 10th century, Cordoba was the capital of the Caliphate, Al-Andalus, that was covering the whole Iberian Peninsula. And Cordoba, by that time, was the largest and wealthiest city in all Europe, just at the level of Byzantium in the east and Baghdad. You remember the so if you look, the most historic remains look quite Arabic in Cordoba. So we have a continuous movement of civilizations. Since then, Cordoba is just a small town with not particularly economical or interest, a part of the touristic attraction and the agricultural sciences. So it's the spring is beautiful. There is a lot of touristic attractions and festivities during spring. We are, you know something about, we are typical flamenco area, but unfortunately that is still also our blooming and harvest season, where we are very busy and we cannot follow that. But this is just to present you Cordoba and to invite you if you don't know the area. Then coming back to legumes, the situation in Spain that somehow parallels that of Europe is that, looking the trend in the last 60 years, the legume cultivation and production was down, till now is stabilized and then is recovering. The situation was that in 1963, this is FAO data, first data that are available on internet is this. So by that time, legumes were very important for human consumption, where we were eating 13 kilos per person year and 20 kilos of meat. 50 years later, the human consumption has decreased markedly by 3, whereas the meat consumption has multiplied by 3. And that is the real situation. Just based as a result of this, legume cultivation decreased, import of soybean increased markedly, as for all Europe, and probably this is just the result of the wealthier economy that changes the food habits, more meat, so then more dependent to soya imports, because we were unable to grow or the legumes that we were needing, not only for animal feeding, but also our needs for human food. We still today largely depend on imports. So we still need to import about 70 to 80% of the lentils, chickpea, or beans that we eat. And there is the interest to revert this trend, and this is what commenting. You might know that the economical situation in Spain was particularly difficult, so that this picture reflects this situation in the 60s. This has changed terribly. Just by curiosity, this was me, this was my family, this was the animal, the tractor, and this is what we had. But well, I mean, this was a particularly difficult region in the mountains of the south, but this was the reality in Spain after several decades after the Civil War. Fortunately, the situation has changed completely. So the situation is that legumes had the consumer had a grown perception that legumes were the food of the poor and they wanted to eat meat. Fortunately, this situation is changing completely today, basically based on consumer perception on health and environmental concerns. Legumes are good for the environment. I don't need to repeat this. I don't have the time that they require less to produce a kilo of protein. Vegetable protein is it take much less resources in water, soil, whatever, than to produce meat. It's very good to have meat. I love meat, but with some legumes from time to time. So this is the situation. So as you can see in the situation in Europe, there is a very recent exponential demand on, for instance, legume flour or meal that was not happening before. So this is a flourishing industry, and we need to provide the raw material for this. The true situation, let's say at the European level now, as I mentioned before, is that pea cultivation in tons produced decreased markedly for a period, but now is significantly recovering. Even soybean, that was never a significant crop, is in the last 10 years markedly increasing, whereas fava bean is just slowly increasing, and unfortunately all other crops like lupins or other legumes like grass peas, the cultivation is still very, very low. The problem is that legume, as I mentioned very quickly before, incorporated to the modern agriculture in Europe did not incorporate well. So just to give you an impression, this is actual data of Spain with average yields. For instance, the yield of wheat was similar 60 years ago to that of legumes, but due to the plant breeding of the agronomic practices, every year there was a yield increase that made today that the yield of wheat is 3 times higher than that of pea and fava bean. So basically we are growing, 10 years ago we were growing lentils, fava beans, chickpeas, lupins, as we were doing 60 years ago, the same cultivars with little improvement. And this needs investment, and this is the concern that we have, and we need support for this. There was an international brainstorming led by Global Pulse Confederation, in which I participated, and the conclusion was that legumes at the global level were receiving less than 1% in research that of all crops, whether the target was that it should be 10%. That is a global level. At European and Spanish level, the situation was far worse. So the European Union is very much concerned on this, on the dependency of Europe to import proteins, plant protein, and they are very much interested in developing, improving the legume production in Europe, as you have seen in the slides before. As a result of this, there is an actual increase in pea, particularly in soybean, but there is still a program that that has to be implemented, and we are still to see the outcomes. As the result of the limited investment in breeding of legumes, as I said, we don't have sufficiently good cultivars. We are doing better and better, but still there was much more investment in sunflower or in wheat or in other crops, and we are paying the consequences. So we are in urgent need of increasing yield and yield stability and addressing market demands. So we need a good cultivar should satisfy the grower, making the crop cost effective and attractive, good yield, standing ability, adaptation, whatever, but also the consumer. meeting quality requirement for the different types. And just for instance, this is not my specialty. I have been more focused on yield stability and resistant to stresses, as I will mention before, but now we are getting involved also on quality. And just giving P as an example, we say P, but we have many different P types. We have vegetable P, where we have types in where we eat the seeds. So here the color, the flavor, texture is important. Other types in where what we eat is the pods, mangetude types, so parchment, other traits are important, or even, this is very important, for instance, in China, where they eat the leaves in soups. For instance, for dry pea, that has been the most important, let's say, in volume of the crop. The traditional use has been animal feeding, let's say, cheap product. But today, with fractionation, separating the protein, the starch, this is becoming very trendy product in human food, and is making the product very interesting, the crop more interesting to farmers. And we shouldn't forget about forage pea, important for biomass, for feeding the animals directly with the hay. For each type, there are different quality requirements, and the good thing is that research has identified sufficient variation for most of these crops, and even at times markers, molecular markers or markers to assist the selection. As I said, my major specialty was not this one, but resistant to stresses. And in P, we have been working, unfortunately, we cannot say we work with one. You need to develop a crop, you need to cover all the needs. So we have been working with too many stresses at different intensities. We just do our best. And not only in pea, but also in fava bean, a little bit, lentil, veggies, chickpea, and even now soybean. And the good thing is that there is progress and sufficient we have huge amount of genetic resources. So resistances to most of the traits have already been identified, or even markers, even in the primary gene pool, but some of them, this was not sufficient, and we need to go to the secondary gene pool, to wild relatives. This complicated the breeding, because the farther you go, the more difficult is across, the worse is the fertility of the progeny, you also introduce undesirable traits, but either in Pisum fulvum or Abyssinicum, resistance has been identified to, and as I will mention in a few examples, not only identified, but also already introduced by crossing into elite pea cultivars. This has been very successful, although slow. So we need new techniques. Now is the second centenary of Mendel, and we remind you that modern genetics started with pea. Mendel discovered the laws of inheritance, and so was very important for the genetics. Unfortunately, in 2 centuries, started very strong, but then the progress was probably slower than in other crops. We lagged behind other crops, but we can say that pea genetics today progressed impressively, and there are plenty of genomic resources available today for breeding. And fortunately, we can say that this is also the case for most legumes. So there are plenty of markers facilitating market-assisted selection. We have in pea tilling population, the reference genome, that is also the case today for even for fava bean this year, for common bean, for lentils, for lupin. So legume has been slow, the progress, but today plenty of resources are available to speed breeding. And just very summary, in very briefly, I cannot go much in detail, but I will try to summarize. in the success from, because we have been working for a big number of very different stresses, from studies at the cellular level, at the plant, molecular, at the field level, till we try to finalize the process of breeding, till releasing of cultivar. So we also do through breeding, and then the commercialization we give to a company. Just giving 2 examples of the cultivars recently released resistant to broom rate that I will mention, or to powdery mildew, this resistant introduced from Python fulvum, from wild relatives. So just I can give you, so also we have to do also the validation in the field, so the yield in comparing in where the red is our new cultivars compared with the standard one when the disease is present. But the good thing is that when the disease is not present, they should also be good, and this is the case also. Just I will give you more on a stress that in which I am now focusing so much recently, but probably for you in the state this is more exotic. You know, is Orobanche crenata is a parasitic plant that is very important. Let's say is the major constraint for all legumes in the Mediterranean basin. So attached to the root, and the same species attached pea, fava bean, lentil, veggies, chickpea, and just give 2 examples of what we have been done. In fava bean, there was already some activities led mainly from Icarda materials. So in Spain, there was already resistant cultivars. There is chemical control with glyphosate at very low rate, and that has been, let's say, successful, working, but not solving the problem, because there was the resistance was just moderate and very low genetic basis. So basically everything came from one Egyptian accession, and there was a lot of work on biological control, suicidal germination, that were nice promises but never succeeded. So as you see, the seeds are in the soil attached to the roots, and they emerge and they breathe the nutrients from the plant. So just very briefly, all the fact that we and other teams have gone is just to identify a number of QTS, explaining very little variation. So this is not yet at the level of being able to market assisted selection. What we have done, and I'm very proud of this, is that we focus it on a different mechanism of resistance. So we pay attention to here phenotyping. So it's a success of phenotyping. We look it for a session where not inducing the germination of the Orobanche seeds. So just and like this, we are at this level of resistance much higher than the existing one. And just giving you an example of 2 breeding lines, Navillo and Quixote. The good thing is that this mechanism is working for Orobanche crenata, that is the plant, the Orobanche species more widely distributed, but it's also effective against a different species that is occurring as a problem only in Tunisia, Orobanche feti, that is red, and against Orobanche felipanche, synonymous Egyptiaca, that's occurring only in the eastern part of the Mediterranean, but is very aggressive there. So in Greece, Egypt, Israel, that area. So we have a resistance that is effective against 3 different species. We hope that this can help to be more durable, but still just complementing 2 different types of mechanisms. We made crosses of this one with the already Icarda existing one, and now we are at the level of releasing cultivars. In pea, the situation was far more difficult. Simply, there was no resistance at all. We screened all cultivars in the market, and they were completely destroyed. So in collaboration with the state, we received a large gene germ plants collection from Pullman Gene Bank. This was the curator by that time. This was me 25 years ago. And we identified some sources of incomplete resistance that by crossing a selection were able to introduce inbreeding. Then we did, I don't know, I cannot summarize here, more than 100 papers on resistance to Orobanche. But in summary, again, as for Fabavin, just small QTLs, explaining a little bit the variation, not sufficient to do marker-assisted selection. We did a gene expression, proteomic, some candidate genes identified, but still not helping the breeding. We did a lot of cytochemical studies, understanding the mechanism of resistance. I cannot go into detail on this. Just the breeding purpose is just, let's say, in this period, first we succeeded identifying sources of resistance and made crosses, making emphasis that the sources, the resistance were in wild types. Then during 10 years, we generated the first generation of very resistant accessions, but still economically poor. And then we focus every year crossing and selection, making sure that the level of infestation was good enough as to not select escapes. And then we have now cultivars that are resistant. It's not complete resistant. You can always have some infection, but that they are sufficiently resistant and have very good standing ability and productivity. So with this, I said, well, okay, I am done with Orobanche. So I am not focusing on this problem anymore. I hope that there are younger people in my institute that will continue with this. So we now focus on other diseases. Just to mention, for Orobanche, but for all others, we are mainly working on resistance, developing crops that can defend themselves. But as of most of the cases, resistance is incomplete. We also do some studies on allelopathy and other crops. Just for instance, here are a number of papers on Orobanche looking for the inhibitory effect of company crops in intercropping, and even with the help of organic chemists, identified the molecules responsible for the allelopathy. I cannot go in detail. Even we tried to do a little bit of biological control, because the problem was so big and so difficult to solve that I was desperate. And already in 1999, I wrote a book chapter on giving recipes on how to eat brown red. So if you cannot fight your enemy, eat it. So we could be a kind of biological control agents. Just to give you another example, I have been focusing on Orobanche because it was a big problem in which there was the need. Nothing was done in pea. I didn't mention, but we worked also a little bit in lentils and in other legumes, Orobanche, but my favorite disease was all been rust. I did my PhD in Holland in resistance to rust, to Puccinia in barley and in wheat. And so we started to work also in rust in Fabavin, Uromyces viciafabe. When we started, there were only levels of partial resistance, incomplete, based on low progress of the disease. We were able to identify by the first time hypersensitive resistance, and so this allowed us to develop for the first time a differential set, something that is done for most crops, but for in Fabavin, pea or lentil, nothing was known whether there were races or not. So we worked on that, and then this allowed us also to identify single genes responsible for the resistance and identify markers. This also we stopped it due to lack of resources some years ago, Fabavin, but I very much would like to continue with this. And then we focus on pea, where we have pea. Everybody says that is Uromyces viciafabe that is infecting pea, but it's true. Uromyces viciafabe can also infect pea, but in temperate climates is mainly Uromyces piscis, and we work on the two of them. and realized that Uromyces biciafaba, in fact, is a complex species. So there are, let's say, forma specialis, isolates of Uromyces. So the rash of faba bean is not infecting lentil and the other way around, or vetch even. So the rash Uromyces biciafabe from common vetch, from Vicia sativa, does not infect lentil and does not infect faba bean. Can infect a little bit, but there is a very nice cross-inoculation example that I am not presenting here. So during some years, we have been identifying, looking for resistance to Uromyces spicy in P, and we went just at the level of identifying good yields. And only this year, we have identified for the first time hypersensitive resistance, not very strong, intermediate one, that will allow us to work more on this. There is work on progress, just at the level of statistical analysis, just the models now. So all the experimentation has been completed on GWAS for resistance to Uromyces spicy different isolates and in the field, in P, RNA-seq analysis, and genomic selection. But this is, as I said, because we are interested, but nobody was very much interested in PRAS, apart from the Australians, that to my surprise has been the only sponsors that I managed to get the Australian farmers that do not have PRAS or lentil RAS, but they were interested in getting ready. So we got some resources that allowed us to do a little bit of work on this. We have also worked on RAS in chickpea, on lentil, on veggies, common bean, and more importantly, most recently, a lot on Latirus, both Cicer and Sativus, doing even QTL, GWAS, and trichotomy, this in collaboration with very nice colleagues of Lisbon, Portugal, Carlota Vaspato and colleagues. Powdery mildew, we work also a lot, and here, but I will just mention, focus on the success that we were able to identify, to discover a new gene in Pisum fulvum and to introduce them into P cultivars very easily. So there were before year 1 and year 2 only that are recessive genes, and we identified the dominant gene year 3 in Pisum fulvum that we introduced. And because most of the fulvum accessions are very, very resistant, even we identified some scar markers, and these need to be, the markers need to be refined now. We want to combine the different genes in a single cultivar, and we are, as I said, we succeeded on this up for a time. This has been stopped. Now we are in the process of analyzing RNA-seq studies and complete GWAS, both to Recife PC and Recife trifolium. Because I mentioned most of the breeding programs rely on year 1 that is considered durable. What is true is an MLO type, but Recife trifolium, the powdery mildew that can infect trifolium, even lentils, can defeat year 1. And we have discovered that. There are studies on this. Similarly, we go to Ascochyta blight, where there was very little resistance. So we screened a very large collection that results can be summarized here. So in most P cultivars, in a scale from 0 to 5, the most resistant that we have in the market is 3. Most of the accessions are close to 5. Only a few wild accessions has less than 3, but particularly resistant. We identified a few ones that were particularly resistant. Contrary, I must confess, contrary to brown rape and to powdery mildew, where we succeeded to transferring this into commercial cultivar, we did not succeed yet to Ascochyta. So what we have is still at the level of the most resistant checks that were already in the market. So let's say a 3. We have done a lot of interesting QTL studies, even proteomics, genomics, different studies, even very recent MACE study identifying candidate genes, but at the level of true breeding, we are failing to go as far as we went for bloom rate and powdery mildew. So till now, I just mentioned a sample of foliar diseases in which we have been working during 20 years. So I am summarizing 20 years of research. But now when going organic, we are having the request to focus deal also with soil-borne diseases, different from bloom rate. That is just a local peculiarity. So we are, and particularly insect pests. The organic sector has a major concern on resistance to aphids and to weevils. And we have done something. We cannot do everything, but we try. And with that said, when we talk about soil-borne diseases, most people think in aphanomyces, what is very important in more humid areas. We don't have aphanomyces in our region. It's very dry and warm, so we have more fusarium wilt, but still under control conditions, we are starting to work with aphanomyces. But we work it a lot. Recently, in the last, let's say, 10 years, started work that just will summarize that we did a PhD with an Algerian student that was with us, so did the PhD on resistance to fusarium wilt in pea, another PhD on resistant in lentils from an Egyptian Iranian student that was with us. We cover from identification of resistance, histology, races, variation in the pathogen, and just occasional studies in collaboration with our colleagues from Portugal in Lisbon, another PhD on resistance to Fusarium Oxysporum special fasciolis in common bean, and more recently in Latidus sativus in grass pea, also resistant. Very interesting studies, very clever and nice student that I am very happy and proud to have the opportunity to work with them. As I said, we are also asked to do something on insects because this is a problem that can be controlled with pesticide, but we need to reduce the input of pesticides. So we did another PhD on resistance to Brutus pisorum in pea, and we're identifying sources of resistance, even going at the level of identification of QTLs, and also on resistance to aphids. Again, even going at the level of identifying some QTLs. Contrary to what I mentioned, pea, resistant in pea to powdery mildew and to brown rape, for all this fusarium and insects, we have been unable till now to do some actual breeding, but we are determined to focus on this. But of course, this will depend on the sponsors. Just as a curiosity, we are more recently, as I mentioned, soybean we are depending of imports. Soybean today, in the last 10 years, increased very marked exponentially in Europe, but in the countries where there is some rain in summer, not Spain. Spain, as I said before, is basically very dry summer, so we can grow very nicely soybean when there is irrigation. Water is time more expensive and more scarce. So I just thought in developing a crop during winter. So our winter is very, very mild. So just developing a crop that can be planting in February and harvest by the late June. And we are making selections. There was very few starting funding accession that could do under this condition, but we identified a few of them, and with crosses and selections, we are working on this. We shall see, because for this we never had a project, but we are interested. Also, we paid a little bit attention in pasture crops, like for instance the annual medics. When we talk about medics, everybody is thinking alfalfa, Medicago sativa. But we in the Mediterranean we have annual species. The most famous is Medicago truncatula, because it has been the model crops in legumes, but it's also a pasture crop. And we have been selecting and trying to develop cultivars on this. But as I said, I don't have much time to go in detail, just would like to take the opportunity to introduce you to the International Legume Society, and that organize a series of triennial conferences, and just mention that the next one is next year in Granada. It's not Cordoba. I never wanted to organize. So it's in Granada, a town, let's say 2 hours from Cordoba. So hope to see you in September next year in Granada. Thank you very much. Just an example of our legume crops, our legume trials, just showing this is very nearby Cordoba, a village with under a castle. So it's a very interesting and historically interesting region. Thank you very much. Just a series of the projects and the actual team, mainly very motivated students that are helping me with all this. Thank you.
Thank you so much, Dr. Diego. It was so pleasure to have all the information from your group. I know you have been working on several crops, and it's so interesting to see what's going on and learning new diseases. And I am so thankful for you to be here today. There are several questions, but I want to start with the question about our collaboration and ongoing world issues. One of your slides, you show us like the meat consumption was increased from 20 kilograms per person for 60. That is quite a bit. And then the legume consumption actually, I think it decreased by more than 60%. So as a breeder, as a well-organized center breeder in Spain, so what are the strategies to have in your breeding program to increase the human consumption as well as the cultivars ready for the drought tolerance?
Those figures are from 2013, so 10 years ago. I didn't show that the trend has reverted since then, and now is increasing. So was decreasing the consumption of legumes till then started to grow again. But the important thing is that even when the consumption was that low, because eating legume was not popular for young people, now it's reverted. Even by that time, we were unable to satisfy the demand. So even by that time, we were importing 80% of the lentils that we were eating are imported from Canada, mainly on North America. About 70-80% of the common beans are imported from Argentina. The chickpeas are imported from Mexico. So I have nothing against global market, but I truly believe that we need to grow legumes for the benefits to the agriculture.
I think so.
I didn't mention, but it was already Columella and Roman, so 2,000 years ago, wrote about the benefits of growing legumes for of course, they didn't know by that time about rhizobium or whatever reason was, but they knew that growing legumes, then the following wheat was doing better. And we need to grow legumes. So to me, that is done by others. But was not the nutritionist, so all the the doctors and medical doctors and nutritionists have been telling us that we were eating more than double meat that was good for our health and less than half legume that was good. But nobody was listening.
Yeah.
But fortunately, in the last 10 years, due to the environmental consent, climate change, this message is, I noticed, is got into the young people, and there is a demand now. So this is what I wrote in my slide. Legumes are becoming trendy again. I don't trust, on the beyond burgers, so I don't think the solution is to produce hamburgers made of vegetable protein, because there is a lot of transformation in there. We have so rich tradition of dishes with soup with lentils of chickpeas, very cheap, very healthy, very nutritious. That is what I trust. But of course, if someone wants to hamburgers, whatever, they pay for that. So
That's
the
absolutely
demand is there. The
yeah.
demand is there and is increasing markedly.
Right. This is the same with US as well. Demand is increasing, the production also need to be increased, but also consumer need to be aware. So our breeding programs more
But
for yes.
but our problem in Europe is that we acted on the demand, and the demand, just the result of the increased demand was importing more, not producing more. And this is what we need to revert, because then we are doing a very good business from the Canadians, so they sell more lentils to us. You sell more soybean for our animal, or the Argentinians sell more common beans. We need to grow more, nothing against market, but simply just to have more legumes in the rotation.
Exactly, that's right, for the soil health and the climate change. So I'm going to go ask you a little bit of specific questions from the breeders that ask questions about. One of the questions is, please briefly describe approximate dates of planting, the planting date and the harvesting time of your dry peas.
For instance, we already planted.
Oh yeah.
Already planted last week the fava beans, the peas. So the planting most of the what we call temperate legumes, so pea, lentil, fava bean, we plant, let's say, from November till January, depending on the region, whether there is rain or not, and we harvest from May till June.
Right. Yeah, it's a kind of little similar. I don't go out to the field until middle of the winter, January, because our temperatures are a little bit warmer than yours. But yeah, I know you already planted in November.
And only the soybean and the common bean, so what we call the summer crops. So we plant somehow by April, or the soybean a little bit later, and then are harvested during the winter, or the summer, at the end of summer.
So talking about soybean is a warm season crop. And how did you adapt that into being a winter crop?
So as I said, we are just paying attention now to the degree days and to the photo period. And just in summary, we, from more than 500 accessions, we were able to select 5 to 10, no more, that we're able to do under those conditions.
Right.
The important thing was our winter is very mild, so the average temperature in January, February is above 10 degrees Celsius. So it's just selection of genotypes that were able to germinate and grow under this low temperature, and more importantly, to flower. So it's a question of photoperiod, that were able to flower by beginning of May, and to set pods during May, June, So there was genetic variation for that, and breeding is just a question of identifying genetic variation and phenotyping and having a clear target. So our target was very clear. I didn't, from 500 accessions, I just throw it away 99% and just retain it, the 1% that was able to do, and crossing and selection. I don't know how far we will be able to do because we don't have a project for that.
Yeah, yeah.
But I think it's promising, and I think it's what we need. And so we can grow very nicely, a very nice soybean crop in the river valleys. We have very good agricultural production, similar to your California, with oranges, with cotton, but when we have water to irrigate. And the problem is that this acreage is limited, the irrigation where we can irrigate, and if we grow soybean there, it's in competition with the maize, because the maize we have to grow under irrigation as well. And
yeah. Here's a good question for you from the audience. Stress resistant is the success of your breeding strategy, perhaps more than due to genetic tool availability. And do you think conventional breeding approaches have an edge that is solely based on the genomic tools?
pardon, which question is that?
This is the question about, I was thinking maybe like you can explain a little bit about the conventional breeding approaches that you have been used for the disease resistant. Is this more helpful for you to have a phenotyping tools or the genotyping tools? Is that?
Yeah, and the point is, I am supposed to be a very good scientist, and I am in a top institute with all facilities. So in my institute, there are all facilities, very good facilities for genotyping and for phenotyping. So there is very good team with sensor, remote sensing, whatever. But I am ashamed to do that. In practice, I don't use that.
Okay.
We are not yet at the level of using this. And my program is so, my applied program, unfortunately, is so small that I, even when we have all types of drones and even a tractor robotic with all kinds of sensors, sorry, I cannot use that. So I just go to the field and this is my sensor, the breeder eyes, and I do selection, visual selection, and the same for the genomic tools. I try to show that we have tried to do kind of identification of markers, now metabolomics, proteomics, genomics. It's very interesting. We are on that, but at the field breeding, what has been successful so far is the classical breeding. That really, I hope that we will be able to use these tools that in which we are working. But as you see, the picture is just me with a few students, and we cannot do more.
Yeah, I agree with you. And you are experienced pulse breeder, and I have been working with a lot of pulse breeders. And yeah, still our eyes and our hands are the best phenotyping tool for us to recognize, especially in the diseases and things like that. Since we are getting closer to finishing up, I wanted to catch up for one question from one of the PhD students. And she's wondering, does the parasitic pathogen, the parasitic weed, grow in faba bean due to common root chemical secretion from the host plant? She is wondering whether the attraction of this parasitic weed is this because of the secretion of a chemical from a root.
Yes, and there is a lot of research on that. The point is that it's basically strigolactones.
Oh, yes.
You see the history. So the point is that the brown rape, and other parasitic weeds like striga, that is more important in sub-Saharan Africa, on maize and sorghum and other cereals, even rice. The parasitic weed has adapted to recognize exudates from the host. So the weeds are in the soil and germinate only when they notice that the root of the host is present. So otherwise they stay there for years waiting. So what is this? It's just a kind of, and then only, well, more recently the scientific community discovered that it's just a question of opportunism. So when there is in the case of the legume, when the crop is having some phosphorus hunger, so has nutrient deficiency, produce more strigolactones that are exudated to the soil, and then the mycorrhizae fungi. So it's what was called before the arbuscular mycorrhiza branching factor.
Yes.
And they discovered that was strigolactones, mainly, many different strigolactones. So what happened? The plants produce strigolactones to facilitate mycorrhization, and Orobanche has taken the opportunity to profit from that, because what would be the reason for a plant to stimulate and parasite? Simply the parasite adapted to this process, so adapted to being able to recognize the strigolactones that are produced by the plant to facilitate the mycorrhization.
Yeah.
And then that's why, so coming back to your previous question, so the good thing will be to develop a model to being able to engineer or to identify plants that do not produce the strigolactones.
Right.
But that we don't have the past capability. We have done it under retrospective, so we have been able to identify plants that do not induce the germination, and then I didn't mention, but there was a postdoc that went to Japan, where they were very good in strigolactone. I identified that in those accessions, flavavin that I mentioned that do not, so chemical analysis with the Japanese demonstrated that they do not produce the major strigolactones that are produced by flavavin.
Yeah.
But strigolactones are needed for proper mycorrhization and branching plant architecture. But there are many different strigolactones, so the strategy should be to engineer a plant that produce at least one or two strigolactones, but not the ones that are recognized by the brown rape, by the Orobanche.
Right.
That is the theory, but in practice, we were able to identify by standard phenotyping a plant that was good branching and was resistant, and then we learned that was because they did not produce the orobancol, the two strigolactone required by Orobanche crenata. But we hope that was producing a third or fourth one that is not recognized by the Orobanche, but is helping the plant to have a good architecture.
Thank you so much there are many questions, but we didn't have much time to discuss all the questions, but we are expecting your arrival here at the South Carolina sometime in the spring, and so the students will be loved to discuss more on diseases and work with you on this parasite weed especially. And thank you, Dr. Diago, joining today for the Going Organic seminar series. And it was really, really eye-opening for all the diseases that we have to get ready for US, and some of the diseases we haven't even seen. So I'm so excited to have your knowledge and share with our audience. And thank you so much. And thank you everyone for joining today. And our next speaker would be Dr. Diago. You need to stop sharing the screen so then we could show the. Okay, thank you so much. Our next speaker would be January 18th. We are going to talk about how to create our soil be more healthy for organic system. Dr. Jeff Shano from University of Saskatchewan, he's a professor. He will be talking on January 18th. Actually, he was one of my professors. So I'm so excited to have him for in the new year. Thank you, Dr. Diago, and we will be very grateful for your appearance today. Thank you so much.
Thank you to you. Bye.